we've got Linxi, and she's been looking at pre-harvest application of sanitisers on high-risk crops to improve food safety and quality. And so without further ado, I'll pass on to Linxi.
Okay. Good afternoon, everyone. I'm very glad to give you a presentation today. My name is Linxi Huang. My supervisors are Professor Zhong Bowman and PhD candidate Hong Shanshang. My honors project is on pre-harvest applications to improve food safety and quality of leafy green vegetables. This is the outline of my presentation. Let's start with introduction. Leafy green vegetables have been considered as one of the crucial components of a healthy diet over the last few years due to its nutritional qualities and convenience. However, there has been corresponding increase in the number of food safety incidents related to consumption of leafy green vegetables since they are usually consumed with less processed. In the past few years, an increasing number of foodborne disease outbreaks caused by LGVS were reported globally. Previous research showed the most common pathogens related to outbreaks of LGVS were mainly E. coli, Salmonella, and L. monocytogenes. From farm to fork, pollution can happen at any time, as microbial contamination of fresh vegetables mainly occurs at the pre-harvest stage. My project is primarily focused on pre-harvest applications. Here are some examples of current control measures. However, more research is also needed to create safe and effective control methods in the future. To improve food safety and quality of vegetables, the industry has used many decontamination techniques. Electrolyzed water, tsunami on farm are widely used in the post-harvest stage, though there is limited information available on the use of nylate, so their effectiveness at the pre-harvest stage requires to be proven. As for innovations, many previous studies focused on the post-harvest application rather than the pre-harvest process. Second, information on the resistance of a pair of E. coli and L. monocytogenes to different sanitizers are also limited. In addition, using nylate as a sanitizer to inactivate pathogens on vegetables was not well documented. The main objective of this study is to determine whether PAA nylate and e-water could be an effective way to inactivate E. coli and L. monocytogenes on leafy green vegetables during pre-harvest stages. The most resistant isolates survival against sanitizers from experiment 1 will be tested by spraying pathogen cell suspensions onto leaves. Since plant quality is also very important, it's necessary to investigate the impact of sanitizers on the plant microbiomes, including both philosophy, bacteria, and fungi communities. We have designed 3 experiments to achieve the objectives of my project. In experiment 1, 10 strains of E. coli. 10 strains of E. coli and 21 strains of L. monocytogenes were inoculated in TSP. Then suspensions of cultures were exposed to 3 sanitizers for 30 minutes. After serum dilution, the diluted samples were plated on TSA, then incubated for 3 days. As for results, I have studied the most resistant strains of E. coli respectively against nitrate, water, and PAA. The most effective sanitizer against E. coli was PAA, as 90% of the strains were comparatively sensitive. 7 out of strains of E. coli showed sensitivity to water, while only 1 strain was sensitive to nitrate. Here are the most resistant strains of L. monosaccharides respectively against nitrate, water, and PAA. Same as E. coli, the most effective sanitizer against L. monosaccharides was PAA, as 80% of the strains showed sensitivity to PAA. 14 out of 21 strains were sensitive to water, while 9 strains were sensitive to nitrate. PAA is the most effective sanitizer, which could be explained by its mechanisms of action and the differences of microsusceptibility. In addition, PAA also had a lower pH compared to water and nitrate, which may sensitize the outer membrane of microbial cells, allowing active components into cells to enhance the effectiveness of PAA. In experiment 2, I tested the 6 most resistant strains and used the same concentrations as experiment 1. The difference is that I sprayed the resistant pathogens on Mizuna. After dilution, I plated on TSA and selective medium Petri film and Paucan agar to determine the effectiveness of sanitizers. I also prepared samples for DNA analysis, but due to time limitation, I haven't got the result yet. Compared to the log reductions of experiment 1, the log reductions of all the strains were less than 3 logs, which indicated that the bacteria are probably protected by the surfaces of plants. Therefore, sanitizers didn't have too much access to them. A higher log reduction of E. coli on Mizuna was observed after treatment with PAA for a longer exposure time. The result was consistent with experiment 1 that PAA was the most effective sanitizer. However, a higher log reduction of L. monosaccharides was found after exposure for 30 minutes, which could be explained that L. monosaccharides may recover after a longer exposure time. In experiment 3, I determined the change of microbial communities of 3 plants without inoculated bacteria onto leaves after I sprayed sanitizers. I have 5 replicates for this experiment. I used different concentrations for different sanitizers on 3 plants based on Hongshan's previous studies. I used 3 different plates, including PDA for fungi and yeast, TSA and R2A for bacteria, in order to engage more different types of bacteria. Then I used the colony lift method for DNA analysis. For experiment 3, the log reduction of sanitizers in activating pathogens on leaves varies between different plants. Log reductions of microbes after 1 week were obviously less than those at immediately harvest, which indicated that the microorganisms may recover after 7 days. In addition, the results also showed that sanitizers work better on fungi and yeast after treatment for 1 week. E-water was more effective for Mizuna and baby rocket immediately after treatment, while PAA worked best after treatment for 1 week. There were no significant differences between isolates from different treatments that were taken immediately at harvest. The untreated samples at 7 days were same at harvest. However, samples treated with water and sanitizers appear to have different communities after 7 days. Theoretically, this could be due to handling the plants like cutting off leaves and the application of water. Overall, I have successfully solved all the research problems. Experiment 1 concluded that the most effective sanitizer against E. coli and air monocytogenes was PAA, as respectively 90% and 80% of the strains were comparatively sensitive. For experiment 2, we know PAA worked best against E. coli inoculated on Mizuna after applying onto leaves. After 2 hours, all the E. coli strains tested were still dying off. E-water worked best against air monocytogenes inoculated on Mizuna after applying onto leaves for 30 minutes. However, it appears that all the air monocytogen strains tested showed evidence of recovery after 2 hours. As for experiment 3, based on the strain survey, there was no evidence that sanitizers impact the microbiomes directly. However, the application of water and sanitizers does after 7 days. The results of this study suggest that harvest should occur quickly after applying sanitizers to avoid recovery of pathogens. Recommended sanitizers appear to be PAA or E-water used at tested concentrations in experiment 3. However, further research is also needed, such as field trials, investigation of plant microbiomes with culture-independent molecular analysis, and the effects on shelf life of LGVS. In summary, the results of this study are meaningful and important to the study food safety and quality. At last, I do appreciate Linxi Huang, who has helped me, especially my supervisors John and Hongshan. That's all of my presentation today. Thank you all.